Today I'm going to make another video talking about how your gut is connected to your brain and how your gut can influence your emotional behavior via what appears to be the vagus nerve. We recently did a video on this series, so I thought this was super interesting. This is all fairly new knowledge coming out in only the last few years, and I thought I would give you an example of this. So let's talk about this. First of all, my name is Dr. Mikola Rashik of Neurogenomics, and let's give you a bit of a background so you can understand the topic today. So everyone knows that chronic stress is a risk factor for depression, right? So that is quite obvious. However, you might or might not know that both chronic stress or depression can alter your gut microbiome. So basically, bacterial composition of your gut. All right, so that's fairly easy to understand. And now we're going to introduce a gut and brain connection because there has been a lot of evidence coming out in recent years that clearly point to the fact that your gut microbiome or the bacteria, the type of bacteria that inhabit your gut, can influence the performance of your part of the brain called hippocampus. And that part of the brain is involved in some of your emotional behavior. So it can affect what kind of mood you are in. So let's talk about hippocampus a little bit. It's this small area of your brain that's deep inside your brain, and this part of the brain is involved in memory formation. So, for example, you can convert short-term memory to long-term memory, and you can do that especially when you are forming new memories that are important, and this can happen. It's also involved in spatial awareness or spatial navigation. So that basically means that how you, when you are in a new environment, how you start memorizing the environment so you can navigate through it or basically move around. That's where hippocampus is also how it's involved in that. So special neurons inside your hippocampus are activated when you are in a new environment so that it helps you start mapping that environment so you have an easier time recognizing later on where you are. And it's also involved in emotional processing information. So hippocampus is also intertwined or works closely with other regions of the brain that are involved in emotions, and we'll talk about that as well. So that can help you, for example, attach emotions to important memories. Okay, so that's your hippocampus, and basically it's the hippocampus, or how the function of the hippocampus is affected, is closely tied to person's depression. So what do I mean? So let's break that down. So for example, chronic stress can be linked to the shrinkage of hippocampus. So if you are chronically stressed, you release this hormone, stress hormone called cortisol, and that hormone constant exposure to that hormone can eventually start reducing the size of your hippocampus. And it's fairly well established by now, scientifically established, that depressed people indeed have smaller hippocampus in their brain than regular, say, healthy, non-depressed people. So there's that. Another concept this is related, is neurogeneration. So hippocampus is one of those areas of your brain, one of those few areas of the brain where you can actually still form new neurons throughout your lifetime in your adulthood. So that means that part of the brain, when you are in depressed state, you start reducing that generation of these new neurons. That's also fairly well established, and that could explain the shrinkage as well. Then the next one is the connection between your hippocampus and amygdala, or amygdala. I don't know how to pronounce it, but I did talk about it in a previous video about the gut brain connection. And amygdala is involved in processing some of the emotions, especially fear-based emotions. And it's closely, it's in the region of the brain that's close to hippocampus. They actually communicate with one another. This is partially how you can form those emotional memories, and if that communication is disrupted, it might negate the person who's depressed the ability to form positive memories, and there is like a relapse towards more negative recall of more negative memories. Okay, so that's a failure there. And then finally, last one is neuroinflammation or brain inflammation, and it is also fairly well established that that inflammation of the brain is connected to depression as well. And again, it's that hippocampus area that can be inflamed. And this is all so much so well established that that hippocampus is involved in the development of depression. That, for example, antidepressants such as the selective serotonin reuptake inhibitors, SSRIs, they are believed, or they're thought to help you deal with depression by stimulating that neurogeneration in hippocampus. And the same thing goes for exercise. We did a video where we looked at what is the clinically established evidence showing how exercise can help you deal with depression and basically help you reduce depressive state. And that is partially because of the fact that it can help stimulate that neurogeneration inside the brain, okay, inside hippocampus. All right, so now what did those authors do? So they've actually done some work in the past. So they in 2020, the same authors showed that they could chronically stress mice. So they basically they would randomly mildly chronically stress mice for a certain period of time, and we'll talk about that in a moment. That would lead to change in the gut composition of the bacteria, change in the bacteria gut composition, and that in turn affected the function of hippocampus, and it resulted in depressive state of the mice that they tested. So they've clearly established that in the past. But finally, we're going to bring out the vagus. Oh, and I forgot to mention, by the way, this video is sponsored by a device that is medically certified, or certified for medical use to activate your vagus nerve. And this is to help you deal with such situations as depression. I've done many videos on this topic, so please check it out. The link to purchase of this device at a discount is available in the video description below. So please check that out if you're interested. So and why do I mention this? Because now I'm going to bring up the vagus nerve. So why were those authors interested in this connection? And that's for a few reasons. Well, number one, vagus nerve is known to innervate a region of your brain, and this is in brainstem, called dorsal raphe nucleus. And that region of the brain sprouts the neurons that produce serotonin, and that those neurons will, like tentacles, shoot out to different areas of your brain. And basically, what happens is, this is basically where how different regions of the brain will receive their serotonin. Okay, and vagus nerve can influence that. In fact, it is also known from the past that if you activate vagus nerve in like the good stimulatory fashion, then you can elevate the serotonin production. I've done a series of videos just on the vagus nerve neuromodulation and how that can benefit dealing with depression. So check those out as well. So that has been established before. So what the authors wanted to know now is whether this connection between the gut microbiome composition and how it affects your depressive state, whether that communication from the gut to your hippocampus, whether you need vagus nerve for that, because vagus nerve does not directly reach hippocampus. It's the serotonin neurons from that dorsal raphe nucleus that reach the hippocampus. So vagus nerve touches that, then that element of the brain sends out its serotonin or serotonergic neurons to hippocampus, and this is how you could be activating hippocampus indirectly via the vagus nerve there. Okay, so they wanted to know, is vagus nerve necessary for this communication? So they repeated the same experiments that I just described, but they did something else. And by the way, this is a trigger warning, because some of the information that I'll describe what they did with the mice is, well, simply might sound unpleasant to some of you. So watch out. Basically, first of all, these mice, all of the mice in this experiment are eventually sacrificed. So they are obviously, they're killed eventually because their brains are being studied. So number one, and number two, trigger warning, because they were chronically stressed. And so let me tell you what they did, and you tell me what you think of that. This is unfortunately how we still conduct experiments, and I'm not saying that I agree with this, but I'm just recounting this to you as to what was done in order to understand what we're understanding now in terms of gut-brain connection. So what did they do is, for 8 weeks, they mildly stressed these mice twice a day. And what did they do? They randomly did number of different stressors. And this included shaking the cage for a few minutes, or tilting the cage at 45-degree angle for a couple hours, making the cage wet. Another one was exposing mice to light at night, or restraining the mice in one position for, say, 30 minutes. Another one was frequently changing the cage for mice, and they are, one more thing that I probably forgot something. One more thing, they also exposed these mice to predatory odors, and that included, for example, rat or fox pee. So all of these, basically, what that resulted is in these mice being stressed, and because they were stressed, it changed their microbiome, the gut microbiome. But they did something different this time, and this was really amazing. They took fecal samples, so poop of these mice, and that, of course, would have the representation of the microbiome from the gut, and they cleaned that up and they fed it to normal, healthy mice. So they put a tube in these mice and they put it directly in their stomach, and basically they seeded the microbiome from stressed mice into normal, healthy mice, and they wanted to see, could the microbiome alone, just the bacteria in mice that have never been stressed, could that replicate those results as if they were actually stressed themselves? And in essence, they did. This is what they did. The first thing they wanted to show is whether the microbiome they put in these healthy mice, never stressed mice, whether that would activate the vagus nerve. And they looked at one specific thing. They basically looked at whether this element of your brain called nucleus of solitary tract, whether that's activated. Why? Because that's innervated by the vagus nerve. I explained that in one of my videos on how the vagus nerve can affect your heart rate. So please check that out. And basically, they looked for a specific protein called CFOS, whether that protein was increased in its amount. Why? Because it's a marker of nerve activation. When nerves are activated, they produce lots of this protein, because this protein is needed for activation of some of the immediate genes. So when nerves are activated and they need to respond, this is one of the proteins that is immediately produced, and they looked for it. They looked for it with antibodies that would attach to this protein that would fluoresce red, and they basically, they looked at mice that were fed this microbiome from unstressed mice, and when they did this, basically you don't see this activation in this nucleus of the solitary tract part of the midbrain of these mice. There is no activation. So basically, this is a standard that the vagus nerve was not activated, versus the mice, healthy mice that were fed microbiome from stressed mice. You could see there is an immediate big red flash there in this area of the brain, meaning the vagus nerve was activated. They also showed that the same region of the brain, the enzymes needed to produce serotonin and dopamine were dramatically reduced, or were reduced. So then they wanted to see, okay, so this shows you what's happening via the vagus nerve, but does that extend to the hippocampus? And they measured that as well. And indeed, they showed the same thing, that when the vagus nerve was activated and you saw this observation of what I just described, hippocampus was also showing elevated levels of that C-FOS protein, meaning the hippocampus nerves were activated, but they showed something else as well. They showed reduction of these specific neurons that they labeled DCX, which are the neurons involved in formation of new neurons. So this is basically they are basically used as a marker of neurogenesis. Okay, and that was reduced in the hippocampus of prior healthy mice that were just fed microbiome from stressed mice, and the hippocampus also showed reduced enzymes needed for production of serotonin and dopamine. So clearly you could see there was this effect on that region of the brain. And the last thing they also showed is that that region of the brain was also inflamed because they measured certain markers of inflammation. So they showed that hippocampus was also undergoing neuroinflammation. So basically what it appears is that when the mice, otherwise healthy mice, were fed the like a microbiome from previously stressed mice, this resulted in activation of the vagus nerve region of the brain that is innervated by the vagus nerve, and inflammation, which we, as I was describing, is involved in the depression of hippocampus, reduced neurogeneration in hippocampus, and of course activation of hippocampus. And then they showed that these mice also exhibited depressed behavior as well, or depressive state. And basically they did variety of tests with these mice. So for example, that are standard for indicating whether mouse is depressed or not. So to give an example, if you give a mouse a solution that contains sucrose versus water, a healthy, excited mouse will go for the sweet pleasure water. Depressed mouse doesn't care. Okay. Another one is, for example, if you hang a mouse from its tail, normal healthy mouse will try to get out of that trap and make itself free, versus depressed mouse will not care, will just hang there. Same thing if you put a mouse in water, healthy mouse will swim around to find a way out, depressed mouse will just float there. And one more thing they did is they also changed the food access. So mice that didn't have food, if you change their cage and then give them food, that healthy mice eventually, while they might be stressed originally because they were in a new cage, eventually they will start feeding themselves because they were hungry. Depressed mice will not show that behavior at the same time span. They will be scared longer, and they'll be too afraid to touch the food. All of this was these mice were affected. So these mice, simply by having microbiome from stressed mouse, they exhibited depressed behavior, which is crazy. To me, I wonder, look, think of it this way. A lot of people don't realize this, they don't think about this, or this concept is not really prevalent in their mindset, that we all have a cloud of microbiome around us that we shed, and that we all shed this microbiome around us, and that we all shed genetic material from ourselves or the microbiome, and we leave that trace behind us all the time. So imagine how easily mice can be inoculated. Now, of course, we're talking about like if you were to go into somebody's microbiome cloud, it's a microdose, and not taking a huge dose like these mouse did. But imagine how powerful just the bacterial composition can be, that if you were to literally eat bacteria composition from a stressed person, or at least if you're a mouse, because all these experiments were done in mice, then you could become depressed as well. All right, now how does the vagus nerve come into this? They repeated all of this, same experiments, but this time they used mice, healthy mice, that had their vagus nerve cut. They literally cut the vagus nerve, so that that transfer of information from the gut to the brain could no longer work, and they wanted to see, could that affect anything. And when they did that, if they fed these previously healthy mice microbiome from chronically stressed, depressed animals, you no longer saw these depressive behaviors in the mice. You no longer saw inflammation of the hippocampus. You no longer saw reduced neurogeneration in the hippocampus, and you no longer saw reduced production or reduced levels of enzymes needed for production of serotonin and dopamine. So basically what this means is that in order to have these effects, in order for your gut to influence your depressive state, you need that in your hippocampus, you still need that vagus nerve communication. So in essence, what these authors showed in this paper is the following, that if you are chronically stressed, this can change your gut microbiome. Again, if you're a mouse, right? And if you change your gut microbiome, this can influence how your hippocampus acts. And based on that, via vagus nerve, couple things are or a few things are happening. Number one, you reduce the serotonin and dopamine production, and number two, you elevate inflammation in the brain, and that inflammation then also can reduce the formation of new neurons in that region of the brain, in that hippocampus region of the brain. Okay. Why this is interesting is also because the authors even mention, well, there's another region of the brain that also can still produce new neurons as well, not too far away from hippocampus. Ooh, I hope I remember the name, called something like subventricular zone. And the reason why I bring it up is because it's known that that part of the brain that also is involved in production of new nerves throughout your lifespan is also involved in your ability to process smell. It is involved in also the acuity, or basically how sharp you are in processing information, and also involved in your memory. And remember, all of these have been deficits described in long COVID. And we did a series showing how vagus nerve neuromodulation could be helping in long COVID as well. So this is an interesting connection. The reason why I bring this up is because it is known that the vagus nerve, while it does not directly influence that subventricular zone, it could affect its function via affecting how new nerves are formed in that zone, via inflammation and via serotonin and dopamine as well. So that's something that is yet very interesting, of something that we should consider maybe exploring. And then, of course, as I mentioned, hippocampus, how your hippocampus performs, that's a target for treatment for depression. I mentioned the antidepressants, or I mentioned, of course, the exercise. But then another option is, of course, and the authors mentioned, is the potential of activating the vagus nerve in a positive fashion. It is known that if you properly activate the vagus nerve, you can stimulate that serotonin production as well. And we did a bunch of videos related to vagus nerve activation and depression as well. So the authors mention this is the vagus nerve. Not only is it an interesting candidate for treatment of depression, it's also an important nerve for helping us understand depression in greater detail as well. All right, that's all I'm gonna have here for you today. Super interesting video, how your gut and brain are connected via the vagus nerve. And I look forward to seeing you guys in the next installment. Bye for now, everyone.